Since the introduction of dual HER2 blockade in the treatment for stage 2 and 3 HER2-positive breast cancer patients, trastuzumab and pertuzumab are combined with different chemotherapy regimens, and the optimal chemotherapy backbone, however, is, well, still unknown, and anthracyclines are still often used. And this, while we know from previous studies, for example, the BCRG-006, that anthracyclines come with a higher risk of cardiac failure, but also a higher risk of secondary malignancies. So the aim of the TRAIN-2 study was to compare the efficacy and safety of an anthracycline-containing neoadjuvant treatment regimen with an anthracycline-free regimen of the same duration, and both in the presence of dual HER2 blockade with trastuzumab and pertuzumab for those stage 2 and 3 HER2-positive breast cancer patients. The TRAIN-2 study was a multicentre randomised phase 3 trial, which was conducted in 37 hospitals across the Netherlands on behalf of the Dutch Breast Cancer Organization. And stage 2 and 3 HER2-positive breast cancer patients were randomised to receive either 9 cycles of PTC plus pertuzumab, which is paclitaxel, carboplatin, trastuzumab, and pertuzumab, or to receive 3 cycles of FAC, trastuzumab, pertuzumab, and then followed by 6 cycles of PTC plus pertuzumab. And all patients underwent surgery after 9 cycles of chemotherapy and completed 1 year of adjuvant trastuzumab. And the primary endpoint of the study was PCR. And this primary endpoint was previously presented at ASCO 2017 and in the Lancet Oncology. And we found high pathological complete response rates for both patients treated with and without anthracyclines. This was around 67%. But we also found more febrile neutropenia and cardiac toxicity in the anthracycline group. And despite these results, not every physician is convinced that anthracyclines can be omitted for all stage 2 and 3 HER2-positive breast cancer patients. And this is because PCR is only a surrogate endpoint for recurrence and long-term survival. And I think it's still a common belief that higher risk patients, for example, with lymph node positive disease, do benefit from anthracyclines. And that is why we present the 3-year follow-up data of the TRAIN-2 study at ASCO 2020, with special interest in the high risk patients. So we included 438 patients in the TRAIN-2 study, 219 per treatment arm, and after a median follow-up of 49 months, we found a 3-year event-free survival percentage of 93.6% in the PTC plus pertuzumab group and 92.7% in the FAC trastuzumab plus pertuzumab group, with a corresponding hazard ratio of 0.9 in favor of the PTC group. And this hazard ratio was not statistically different. And we also found no anthracycline benefit for any of the subgroups, as age or disease stage. And if we look more closely at the higher risk patients, we also found no anthracycline benefit for patients with N2 or N3 lymph node status. And the 3-year estimate overall survival was 98.2% in the PTC plus pertuzumab group and 99.7% in the FAC trastuzumab plus pertuzumab group, with a hazard ratio of 0.91. And this was also not statistically different. Well, safety of the neoadjuvant treatment was previously published, and I mentioned it also before. So we found more febrile neutropenia, but also more hypokalemia in patients treated with anthracyclines, and cardiac toxicity was significantly higher in the anthracycline group, with a left ventricular ejection fraction decline in 36% of the patients treated with anthracyclines and 22% of the patients in the PTC group. And I think what is also a mentionable new finding is that 2 patients in the anthracycline group developed a chemotherapy-related acute leukemia. So I think those are the most important findings. The 3-year follow-up of the TRAIN-2 study shows no event-free or overall survival benefit for use of anthracyclines in patients with stage 2 and 3 HER2-positive breast cancer who received dual HER2 blockade. There's also no evidence that high-risk patients will benefit from anthracyclines. The anthracycline-containing chemotherapy regimen, however, comes with an unfavorable toxicity profile for both short-term and long-term toxicity. And therefore, I think a neoadjuvant anthracycline-free regimen containing carboplatin and taxanes with dual HER2 blockade should be considered in all stage 2 and 3 HER2-positive breast cancer patients. Well, with these great and early responses that we saw in the TRAIN-2 study, we think that one of the next steps is to de-escalate cytotoxic treatment, of course, where possible. So questions that arise are, is a pathologic complete response after, for example, 3 or 6 cycles of chemotherapy, is it as good as reaching a pathological complete response after 9 cycles? So who needs 9 cycles of chemotherapy, and can we use, for example, radiologic response to adjust the number of chemotherapy cycles? So the aim of the TRAIN-3 study is to evaluate efficacy of image-guided de-escalation of neoadjuvant chemotherapy for the same study population, so stage 2 and 3 HER2-positive breast cancer patients. And the TRAIN-3 is currently already enrolling in 50 hospitals in the Netherlands. And such a large study with great impact can only be organized in cooperation with the Dutch Breast Cancer Research Group. And also, of course, with all participating hospitals, doctors, and most importantly, I really want to mention that with all patients that are willing to participate in clinical trials. The most ideal situation, that will be that we will be able to select patients upfront for different treatment strategies. So some patients maybe could be treated totally without chemotherapy, but others might need some other HER2-directed compounds or definitely need chemotherapy. So I think we are in high need for predictive biomarkers to make a better selection of patients in whom we can further de-escalate and in who need other treatment strategies. So also at this moment, we are studying those biomarkers in the TRAIN-2 population, and that's also a big sub-question of the TRAIN-3 study.